So I asked you to bring your lecture notes with you, so that's what we're going to start with. Just one quick announcement before we get started about next week. You guys do have another quiz next week, and that quiz is going to cover cellular respiration and fermentation and cell division. Okay, so cellular respiration and fermentation and cell division. So it won't include cancer or the stuff that we start today in terms of sexual reproduction and meiosis. Okay, so that's what we're starting. We're going to talk about sexual reproduction and meiosis. Just to remind you about something we've already talked about earlier in the course, we mentioned asexual reproduction, and we said that in this form of reproduction you have one parent that gives rise to the offspring, and then some examples of asexual reproduction would be when you have a single-celled eukaryotic organism that divides by mitosis to produce two new single-celled eukaryotic organisms. That would be a form of asexual reproduction. Also binary fission, which we discussed at the beginning of cell division, which is a cell division process that happens in prokaryotic cells, is another example of asexual reproduction. In binary fission, you also start off with one cell divides and gives rise to two daughter cells, that also being a form of asexual reproduction. It's also important to note that in asexual reproduction, the offspring that you produce are in most cases genetically identical to the parent, okay? And that's going to be in contrast to sexual reproduction. So in asexual reproduction, the offspring are in most cases genetically identical to the parent that produced them, okay? In sexual reproduction, that's not true. The offspring are going to be genetically distinct or different from the two parents that produced them. Okay, so sexual reproduction we also discussed at the beginning of the course, and we said that this type of reproduction involves two parents producing the offspring. And then again, the offspring that are produced are genetically different from either parent that produced them. Okay. In order to understand a sexual life cycle, we have to define a few terms. Some of them we already defined, so I'll just mention them and not write them on the board. But first we'll start with germ cells or germ-line cells. And germ cells are the cells that give rise to gametes. Okay, so germ cells are the cells in your body that are going to give rise to gametes. So let's just define what gametes are. So I told you previously that in humans the gametes are sperm and egg. So gametes are haploid reproductive cells. With examples being sperm and egg. So what happens is the germ cells are going to produce the cells that develop into either a sperm cell or an egg cell. Okay, and then also just in terms of haploid versus diploid, germ cells are diploid. Okay, so what we're going to see and what you learned last week happens during meiosis. Is when the cell goes through meiosis, the daughter cells, you start off with a diploid cell and you produce 4 haploid cells. Okay, so this germ cell is going to have to undergo meiosis to produce the haploid cells, and then those haploid cells develop into or differentiate into a sperm or egg depending on the gender of the person. Another thing that you should know is somatic cells. Somatic cells are all the cells of the body except for your germ cells and gametes. So the soma means body. So again, all the cells that make up the body, except for your germ cells and gametes, are called somatic cells. And I told you previously that in humans, somatic cells are diploid. So in humans, your somatic cells are diploid. Okay, and I told you that basically every cell in your body is diploid except for your gametes. Okay. Okay. A zygote, and again in humans, this is going to be diploid, is the cell that's formed by the union of haploid gametes during fertilization. Okay, so what happens? So fertilization simply is when two haploid gametes fuse to form the zygote. That's what happens in fertilization. Okay, so for the process of fertilization is two haploid gametes uniting and producing a zygote. Okay, so the product of fertilization is a zygote. So what happens is you have two haploid cells, and when they unite, they each contribute a set of chromosomes. And so that's why the zygote is diploid, because they got one set from the sperm and one set from the egg. Okay. So all these different types of cells are going to be involved in, well, somatic cells not so much, but germ cells, gametes, and a zygote are somehow involved in the sexual reproductive cycle of animals. Okay, and then just to remind you, we've already discussed haploid versus diploid when we discussed cell division. And remember that haploid refers to having one set of chromosomes, and diploid refers to having two sets of chromosomes. Okay. So the first thing that we're going to go through is the sexual reproductive cycle that most animals undergo, and that includes us. Okay, so we're going to discuss the sexual reproductive cycle that most animals undergo. So it's basically going to follow what's on the board there, but I would just suggest drawing it out in your notes. So this reproductive cycle is going to start with two individuals, okay? And then what happens is germ cells in each of these individuals undergo meiosis to produce haploid cells. So this is the germ cell within this individual undergoing meiosis, a germ cell in this individual undergoing meiosis, and that produces a haploid cell, or haploid cells, and those haploid cells are going to be the gametes. Okay? And then once you get the gametes produced, the next step in the cycle is fertilization. Okay, so during this process, you're going to have two gametes unite. And the product of that is a zygote, which is a diploid cell. So the product of fertilization is a diploid cell called a zygote. Okay, and then to complete the cycle, what happens is this zygote is going to divide through mitosis, and then that produces the multiple cells that are going to make up the body of the offspring that's produced as a result of this reproductive cycle. Okay, so that kind of also puts mitosis and meiosis into perspective with respect to a reproductive cycle. So meiosis produces the gametes in this reproductive cycle, and then mitosis takes you from a single-celled zygote into a multicellular organism. And then obviously more processes in addition to mitosis are involved. You have differentiation and other things occurring to produce a fully formed human. Okay, so I just wanted to point out, we're not going to go through them. If you go on to take 1114, you will learn them. So this is one sexual reproductive cycle, and this is the one that animals, including us, undergo. But there's other reproductive cycles, and one thing they all have in common is that they all include mitosis, meiosis, and fertilization. Okay, so all the reproductive cycles include mitosis, meiosis, and fertilization. It just depends on when they happen within the cycle. Okay, so in 1114, you'll go through a life cycle that most fungi undergo, and in that particular life cycle, the one thing that's different is that the multicellular form of the organism is haploid. Okay, and what happens when you have fertilization producing the zygote in this life cycle is the zygote, instead of undergoing mitosis like it does in this cycle, it undergoes meiosis. Okay, so how the when the different processes take place is going to change depending on what life cycle you're talking about. Also, this is the life cycle that most plants undergo, and there's actually two multicellular forms in this life cycle. There's a diploid multicellular form, and that's the one you actually usually see out in nature. And then there's also a multicellular haploid form of the plant, which is in contrast to ours because we only have single-celled haploid cells in our reproductive cycle. We don't have a multicellular haploid form. Okay, so you do need to know this sexual reproductive cycle, but I just want you to be aware that not all are the same and that they can be different. And you'll learn about these in 1114 again if you go on to take that. Okay, I also wanted to remind you of a couple of things that we talked about before discussing cell division. The first thing is sister chromatids. And just to remind you that sister chromatids are two copies of a duplicated chromosome that are attached to each other. Okay, so you have this single chromosome, and then you're going to replicate it, and then right after you replicate it, the two replicates remain attached to each other for a while, and as long as they're attached to each other, you call them sister chromatids. Okay, and then eventually when you separate them from each other, then they're individual chromosomes again. Okay. During meiosis, it's important to be able to distinguish between what sister chromatids are and what homologous chromosomes are. Okay, so just to remind you that homologous chromosomes are a pair of chromosomes, so it's two chromosomes that carry the same set of genes. Okay, so here they're showing you two chromosomes, a pink one and a blue one. Each chromosome has already been replicated and consists as a pair of sister chromatids, and you can see that the homologous chromosomes carry the same set of genes, so they each carry A, B, and D, but you can see that the homologous chromosomes are not identical to each other because they can carry different alleles for the same gene. So the one example in this picture is that for the B gene, the pink chromosome carries a capital B allele, whereas the blue chromosome carries a lowercase b allele. So again, it's the same gene, but a different form of that same gene. And that's what makes homologous chromosomes different from each other, is the fact that they're going to carry different alleles for particular genes. Okay, but again, the homologous chromosomes are a pair of chromosomes that carry the same set of genes. And in humans, I told you that you actually have a chromosome number 1, and then you have all the way through to a chromosome number 22. And I told you that your somatic cells have 2 copies of every chromosome. And so the 2 copies of chromosome number 1 are homologous chromosomes. The 2 copies of chromosome number 2 are homologous chromosomes, and etc. Okay, so those are the homologous chromosomes in humans. Okay, so what we're going to do is we're going to start by watching this video that describes a little bit about the life cycle that I just discussed, but then it also goes through each of the stages of meiosis. And meiosis actually includes two cell divisions. So it's going to include meiosis 1 and then meiosis 2. And this video will take you through both of those divisions.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, meiosis 1, halves the number of chromosomes, and in the second division, meiosis 2, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis 1 begins with prophase 1. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just as easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has 2 sister chromatids. However, we are separating homologous chromosomes into 2 different cells, so that each new cell will have only 1 member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, 2 cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only 1 of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber. And in telophase 2, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
Okay, so that kind of took you through both cell divisions, meiosis 1 and meiosis 2. And just to compare it again to, we talked about a cell division process called mitosis. In mitosis, let's say if you start off with a diploid cell, and that diploid cell undergoes mitosis, then you're going to produce 2 diploid cells. Okay, similarly, in some organisms, haploid cells undergo mitosis, and if a haploid cell undergoes mitosis, then you get the production of 2 haploid cells. Okay, so mitosis here again. Mitosis produces daughter cells that are genetically identical to the parent and also genetically identical to each other. Okay, so mitosis produces daughter cells that are genetically identical to the parent cell and also genetically identical to each other. In contrast to this, at meiosis, you're going to start with a diploid cell, and this diploid cell is going to undergo the first meiosis, meiosis 1, and that gives rise to 2 haploid cells. So the first division of meiosis halves the number of chromosomes. Okay, and then each of these cells undergo meiosis 2 and produce in total at the end 4 haploid daughter cells. Okay, so in contrast to mitosis, in meiosis you produce 4 daughter cells rather than 2, and then you can also see that the daughter cells are not going to be genetically identical to the parent cell. The most obvious reason for that, or the most obvious from this picture, is the fact that they have half the number of chromosomes as the parent cell. Okay, so that's another thing about meiosis. The daughter cells are going to have half the number of chromosomes as the parent cell, and they're also not genetically distinct from the parent in other ways. They're also genetically distinct from each other. Okay, so none of these haploid daughter cells is genetically identical to any of the other daughter cells. Okay, over here, these 2 daughter cells are genetically identical. These 2 daughter cells are genetically identical, but the 4 daughter cells produced in meiosis are going to be genetically unique. Okay, and the 2 things that cause that are going to be independent assortment and crossing over. Okay, what I said it in opposite order of which they occur. So crossing over happens first, and then independent assortment happens after that. Those 2 processes are going to lead to the daughter cells being genetically unique from each other. Okay, so that's just a useful comparison in terms of the products of mitosis versus the products of meiosis. Okay, and so this is just showing you what I just went through. So similar to mitosis, before you enter meiosis, you're going to replicate all the chromosomes in the cell, and that's going to happen during an interphase that precedes meiosis 1. Okay, so this cell has 2 chromosomes. It replicates both of them. The chromosomes condense, and then what happens in meiosis 1 is these 2 chromosomes here are homologous chromosomes, and in meiosis 1 you separate them into separate cells. Okay, so meiosis 1 separates the homologous chromosomes from each other, and that produces 2 haploid cells, because this is, remember, as long as they're attached, that's only 1 chromosome. And then in the second meiosis, you separate the sister chromatids from each other, and then that's going to produce 4 daughter cells, each which are haploid and only have 1 chromosome. Okay. Okay, so again, the first division, meiosis 1, halves the number of chromosomes. That's also when you separate homologous chromosomes from each other. And then in meiosis 2, that's when you separate the sister chromatids from each other to produce your 4 haploid daughter cells. Okay, and so what we're going to do is we'll stop there for today. And what we're going to do is meiosis 1 and meiosis 2 are both broken down into the same phases we broke down mitosis in. So you break down each one into a prophase, prometaphase, metaphase, anaphase, and telophase. And so similar to the mitosis, we're going to go through and define the events that you need to remember for each of the phases. Okay. Okay, so we'll stop there for today. And then in terms of today's lab, the first